All right, so now we have all of our data that's been corrected for blanks. One thing I do want to do is just add the urea concentrations across the side. It's just going to help keep track of everything as we're going through this. The next step is to create a graph that overlays the emission spectrum of your protein at each urea concentration. So we're trying to create a graph that looks like this, which you can find or something similar as an example in your lab manual. So we'll have more curves than this because we'll overlay all the urea concentrations, not just these two, on the same plot, with fluorescence on this axis and then wavelength over here. So let's go ahead and make plots like this with our data. Chart, scatter, there we go. Just move it over here. And then you'll need to adjust the axis so you can kind of see. And we measured from 300 to 370. Yep, so that should look pretty good. And then here you can see all of the data. All right, so again, we have these curves that correspond to fluorescence signal versus wavelength. So one of the first things that I noticed looking at this graph is that you have this one curve that's pretty much flat down here. So the height of these curves corresponds to basically how much protein you have. Remember, we're using fluorescence of the tryptophan, which we know absorbs at 280. We've used absorbance at 280 before to measure protein concentration, right? You know that that's directly proportional. And so if you have more protein, you're going to see higher signal. Less protein, you're going to see lower signal. So it looks here like there's very little, if any, protein that got into this well. So probably what happened, and you guys know when you were doing this experiment, there's a lot of pipetting of small volumes of things into a bunch of wells. And so depending on how you do that, it can be easy to miss a well or sometimes double up on a well. So you want to watch for that at this step. So particularly if you see a bunch of samples where you have just like flat lines instead of these kinds of curves, you want to be pretty concerned. And in general, you can actually throw those points out. You'll want to state in your methods section something like data from wells that appeared to have no protein was discarded. So that's something that you want to do. So you may be tempted as you're watching this video and thinking about the analysis, like to maybe just plot the A data or the C data and not D and E. But it's actually important to stop and take a look at all of your data so you can see any points where you may have potential problems. So let me do that then for all the other points. So this one, I think you can tell, looks a little bit better. It doesn't have that that place where you're, or that condition where you're missing protein, right? You can see that they all look nice and curvy. Yeah, and then the other thing you should see, right, is a difference between folded and unfolded. So let's take a quick closer look at one of these curves. So remember, what we're trying to do is use the shift in these peaks to measure the folding to unfolding transition. So in looking at our data, we can see a nice peak here corresponding to folded protein, and then a nice peak over here corresponding to unfolded protein. And you can see this shift at the concentrations in between. Another thing to notice about this data is that as the curves are shifting from folded to unfolded protein, there's a place around a wavelength of 340 where the curves kind of cross each other. And there's not a perfect place, but there's sort of minimal difference between all the curves, somewhere right around 340. And we're going to use that intersection wavelength later on for normalization. So this data looks good. Let me quickly plot the third replicate. So here you can see something a little bit strange happened, where there's this huge peak. And again, remember that the height of that peak really just corresponds to the amount of protein you have. So it looks like, again, there was some sort of pipetting error where somehow protein got added multiple times to this well. And so that's why you have this huge peak here. So let's see if the other ones look a little bit better. Yeah, so it seems like in the case of the mutant, across all of your replicates, there's something going on with the amount of protein. So at this point, I would probably go back and think about how this experiment was set up, whether different people pipetted things, or what may have happened that would contribute to this. So here in this one, you can see there's a case where there's no protein here, and extra protein here. But the good news is, this is a pretty good problem to have, actually, because we're going to normalize for protein concentration anyway. So as long as the shift is still there, which it looks like it is, it's a little hard to see because the scale is so different, but as long as the shift is still there, the data is still perfectly usable. So if you're going to make a mistake, protein concentration is an okay one to make.